and he's going to talk us about selecting a predictive and diverse set of SNPs from GWAS data with the help from Hi-C information. We are not able to see the screen. Is it possible? Or it's a PDF this time, not PowerPoint. I'll open the PowerPoint if that's. All right.
Hello everyone. I'm here to present you our project, which is about selecting predictive and diverse SNPs and genome-wide association studies, and the application of the Hi-C data in the context of SNP selection problem in the form of a SNP interaction network. So our purpose is to identify a SNP set which is associated with a phenotype, and this corresponds to the feature selection problem in the machine learning context. There are a few challenges for this problem. First, SNPs are not individually strongly associated with complex phenotypes, which means that the joint effect of multiple SNPs actually explain the phenotype. And there is also the issue of data. We have low sample size with high number of dimensions, high number of SNPs. So our approach to overcome these challenges is to account for the genetic interactions by utilizing a SNP-SNP interaction network, which is constructed using some a priori biological information, and primarily the Hi-C data. In the literature, there is already a method that follows this kind of approach, named SCONS. It employs an additive model and encourages the selection of connected SNPs on the network. This way, it can recover SNPs which are associated with a phenotype, but has not been identified before. The issue, however, is the opportunity cost for the purposes of phenotype prediction. Now, let's look at why. Encouraging the selection of connected SNPs lead to the selection of closed SNPs on the network. And the closed SNPs are actually more likely to carry redundant information because they are more likely to be in highly correlated haplotype blocks, which are inherited together. Secondly, distant SNPs are more likely to affect different biological processes, which means that prioritizing distant SNPs would result in selecting a more complementary SNP subset. So following this intuition, with our method SPADIS, we set out to select a diverse SNP subset by penalizing close proximity on the network. For this purpose, we utilized a total of 4 networks, 3 of which are already existing in the literature introduced by the authors of SCONS. The genomic sequence network connects SNPs that are adjacent on the DNA sequence, while the gene membership network additionally connects SNPs that are near the same gene. The gene interaction network, in addition, connects SNPs that are near interacting genes obtained through a protein interaction network. And we have constructed another network, the 3-dimensional genomic sequence GWAS-ICN network, which connects SNPs that are adjacent on the DNA sequence, as well as the SNPs that are significantly close in 3D. To construct this network, we first process the interchromosomal contact matrices and assess the statistical significance by using the FitIC method. Then, in our network, we connect the SNPs that are significantly close in 3D. Regarding this, there's a study which suggests that strong three-dimensional spatial interactions on gene level indicate similar gene functionality. So following this intuition here we apply it on the SNP level with our network. So the connections that the connection of the SNPs that are adjacent sequentially contains information regarding linkage disequilibrium, while the connections for the closed SNPs in 3D contains information regarding biological function similarity. And with SPADIS, we exploit this information to acquire a complementary SNP subset. Now let's look at how SPADIS operates. SPADIS requires 3 inputs: a genotype data, which contains the number of alternate alleles for the corresponding SNP and for the corresponding samples, and the phenotype data for each sample. And there is the SNP network that we already talked about. Due to the nature of the problem and also the availability of the data, the number of SNPs are much larger than the number of samples. So in SPADIS, SPADIS operates with a 2-step approach. In the first step, a phenotype association score is calculated for each SNP by using the genotype and the phenotype data. We calculate it using the sequence kernel association test, or simply SCAT. In the second step, in the second stage, SPADIS operates with a cardinality constraint and selects a SNP subset, while rewarding phenotype association scores in an additive model and penalizing close proximity on the SNP network. Our optimization function that achieves this have some useful properties that we can utilize, such as submodularity. So now you may wonder, why is this any important? So how is submodularity any useful? Well, the thing is that the subset selection problem with cardinality constraint is NP hard. So we need an approximate algorithm to solve this problem efficiently. And this is where the submodularity property comes into play. There's a theorem which suggests a greedy algorithm that is guaranteed to return a constant factor approximation to the optimal solution for submodular, non-negative, and monotonically non-decreasing functions. We utilize this algorithm, this greedy algorithm, to find a near optimal solution. And since this is a greedy algorithm, it is efficient time-wise. Let's see our results. We benchmark the performance of our method in the dataset for Arabidopsis thaliana with nearly 200 samples and nearly 200,000 SNPs. It has 17 continuous flowering time phenotypes. We first evaluate the performance of SPADIS in phenotype prediction performance by looking at Pearson's squared correlation coefficient, which corresponds to the variance explained in the phenotype. On the figure on the left side, we compare SPADIS with scones and also some other baseline methods. The values here are averages of all 17 phenotypes and all 4 networks. So the best performing methods here is SPADIS, and it is followed by scones. So that's why we performed some more additional tests, some additional experiments for comparing them. And on the figure on the right side, we compare SPADIS and scones over different cardinality constraints. And the figure shows the improvement of SPADIS over scones. So SPADIS provides some consistent improvements in phenotype prediction performance. And we have benchmarked the performance, the CPU runtime performance of the compared methods. The fastest one is univariate linear regression, which is just a simple baseline method that doesn't perform well in phenotype prediction. And it is followed by SPADIS. So SPADIS is faster than scones and all the remaining methods by at least an order of magnitude. And we have evaluated the contribution of the high-C data in phenotype prediction performance. This table shows the Pearson squared correlation coefficient values for different cardinality constraint experiments and for different networks. The best performing network for each experiment is shown with bold text here. And the GS-ICN network is better than all other networks on average in 4 out of 5 cases. So the high-C data provides some consistent improvements, but slight improvements in phenotype prediction performance. So to summarize, SPADIS is a novel SNP selection algorithm that rewards the selection of remotely located SNPs. And it provides improvements in phenotype prediction, and it is efficient in terms of runtime. And we have also investigated the 3-dimensional conformation data, the high-C data, in the context of the SNP selection problem, which yields some slight improvements in phenotype prediction. So it shows promising results, but there is a lot of work to be done to fully utilize it. So thank you all for your attention. I would like to thank my advisors, Özür Taştan and Ercüment Çiçek, for their contributions. And I will be very happy to take any questions and continue this discussion further or over my poster later today.
Are there any questions? Maybe one very quick. Or we are running 15 minutes behind, so I will take it offline. Thanks a lot for the talk. Our next